Hello. Today we are going to talk about thin layer chromatography. When it is about chromatographic separations, this can be achieved by two basic techniques, which can be talked as modes of chromatography. One is column chromatography, and second is thin layer or planar chromatography. In column chromatography, the stationary phase is attached to a suitable matrix and packed in glass or metal column, and the mobile phase passes down the column, either by gravity feed or by pumping or applied gas pressure. The second type is thin layer or planar chromatography, in which the stationary phase is attached to a suitable matrix, is coated thinly onto glass, plastic, or metal foil plate. The mobile liquid phase passes across the thin layer plate, which is either held horizontally or vertically or by capillary action. This mode of chromatography has practical advantages over column chromatography. The thin layer chromatography can be used to analyze a large number of samples simultaneously. Example of thin layer chromatography is silica gel coated glass, plastic, or metal foil plates. Also, the planar chromatography can be described by paper chromatography as an example, in which the stationary liquid phase is supported by cellulose fibers of the paper sheet and uh, the mobile phase runs over it. So, with thin layer chromatography on a plate, the mobile phase is passed over the immobilized stationary phase by capillary action. This form of chromatography is one of the oldest form and has few applications in biochemical analysis. As we see in the slide here, just separation of simple two colors of ink, that is red color ink and blue ink, if we want to attempt in a simple laboratory setup, it can be done by thin layer chromatography. As you are seeing in a plate, it is mixture of red ink and blue ink. Here is a capillary tube held, and here is the paper or TLC plate that is shown. And this is the chromatography system or chromatography chamber, in which isopropyl alcohol and water as a mixture of mobile phase or solvent phase is shown. In the next picture in the slide, you see the capillary is dipped into the sample, which is going to suck the sample. The amount of sample to be loaded is to be very minimum, otherwise, it will bleed and separation of sample analytes cannot be done. Now, in the next picture, what we see is the lid of the chromatography chamber is opened, and then you are going to insert this separation plate or chromatographic plate inside the chromatography chamber. Last is you have to cover the lid and observe the movement of the mobile phase from one end of the sample plate to another end of the sample plate. Of course, there are certain precautions when you are going to load the chromatography plate. As I said, the spot that you are going to place on the chromatography plate should not be big. It should not bleed. It should not diffuse. At the same time, it should be adequate to have enough amount of sample to be loaded. Secondly, the chromatography chamber lid should be kept close. That would allow saturation of chromatographic chamber with the solvent vapors. Insertion of air is going to interfere with the efficiency of the system. The moisture in the air can interfere with movement of analytes on the TLC plate or the paper. So the chromatography chamber must be allowed to sit for some time before we begin with the experiment. The lid should be closed and the chamber should be allowed to saturate with solvent vapors. Once the sample analyte is loaded onto paper or TLC plate and is immersed in the chromatography chamber, the lid is tightly closed, and the solvent front migrates from lower end to upper end. So, in short, when it comes to TLC, it is a method for separating the compounds in the mixture. As we have seen the previous plate, two ink color spots have separated. Now, how much quantity is to be loaded? It should be as small as possible because when it comes to efficiency or sensitivity of TLC technique, microgram quantities also can be analyzed by TLC. It takes little time. It is fast, simple, and quick method, so is a method of choice. At the same time, the efficiency is really high. When it comes to TLC, there are three steps: spotting, development, and visualization. Spotting or loading the sample is the first step, where the sample to be analyzed is dissolved in a volatile, that is easily evaporatable solvent, and a solution, a sample solution or analyte solution, which is dilute enough, is prepared. Spotting consists of using a micropipette or a small glass capillary, which is going to be dipped into the sample, and by capillary action, it is going to hold very small quantity of the analyte, and it is spotted onto TLC plate. TLC plate is powdered silica gel which is coated onto a plastic or a glass sheet. We'll talk about it in the next slides to little more details. And the third step, that is development, it consists of placing the bottom of the TLC plate into the solvent system or the development chamber. Now, when you are going to put your TLC plate into the solvent chamber, you have to take care that the sample loaded should not get merged into the solvent system, otherwise, it will travel into it and disappear. So, the original spot should remain above the level of the solvent system. And the last step in the process, once the development is done, is visualization. So, once the sample has traveled, the analytes are separated, you have to take the plate out, dry it, and visualize it. Visualization in case of red ink and blue ink was easy because the analytes are colored ones. But when it comes to biochemical samples like sugars, fatty acids, etc., it cannot be directly seen. So you need a developer or an agent, a chromophore to be added, which can develop the system. For example, ninhydrin is commonly chosen development solution for detection of amino acids. Now let's look into the summary of what we talked. That is, when it comes to uh, working or using or practicing TLC or its advanced version, that is HPTLC, high performance TLC, the basic steps are sample preparation, sample application, chromatographic development, visualization, and then the scanning and documentation using different advanced computerized systems. As we see in this slide, a chromatography plate is seen. As we see, it is having some labels. This is a starting material and this is a fraction mixture. So when you are going to analyze a particular mixture, you have to place some controls or pure chemicals which are going to work as standards, and based on RF value you can compare the analyte and detect the analyte present. This is the chromatography chamber. As you can see, as many as 10-12 samples can be easily run simultaneously when you are using a chromatography chamber of this size. This small photograph is showing the chromatographic plate. Now, while handling the chromatographic plate, you have to be very cautious that it is to be handled very carefully. Your thumb impressions should not be marked, as the sweat or even the surface of your hand is going to have certain biochemicals in the sweat which are going to pollute the chromatography plate. So it is to be handled only after wearing of by wearing of gloves. Now, when it comes to the principle of thin layer chromatography, evaporation, condensation of mobile phase is one, and adsorption and desorption of the analyte on the stationary phase is another phenomena that is happening. So, when we talk about TLC, the support on the stationary phase is glass or plastic. Glass is commonly chosen. Of course, these days, alumina foils also are available, which are used to coat the silica gel, and ready silica gel plates, which, which use alumina as the base, are available on the market. Adsorbent layers and stationary phases, wherein standard silica, that is silica coating, silica 60, which is referred to as with a pore diameter of 60 angstrom, is most commonly used adsorbent in the TLC. Alumina oxide, cellulose, polyamide are other some materials which are used for coating the support. Of course, selection of an adsorbent is an important step where the solubility of the sample compounds and the interaction of the sample with the silica or the any other support chosen, it is to be analyzed. The hydrophilicity or hydrophobicity of the analyte and chemical reaction with the adsorbent or the eluent is going to decide the efficacy of the process. So the selection of support or the stationary phase is an important step. Simultaneously, the mobile phase which you are going to choose also is very important and needs to be talked about. But before we go to discussion on uh, mobile phase, the basic is to be understood. That is the principle of TLC system. So when the principle of TLC system is to be described, a thin layer of the stationary phase is formed on a suitable flat surface. That is silica gel that we coat onto the glass or alumina or plastic sheet. Since the layer is so thin, the movement of mobile phase across the layer is generally by a simple capillary action. It is rapid and there is very little resistance to the flow. So, as a result, the run of the mobile phase onto the stationary phase is rapid and the entire experiment gets over very fast. So, when it is to the principle of the system, it is about the different affinities for the stationary and mobile phase, that of the analyte. So adsorption capacity of each component in a mixture for the same adsorbent are different. As the mobile phase moves across the layer from one edge to opposite, it is going to transfer the analytes placed. So along with the mobile phase that is running, the analyte also is going to move. But when this movement is happening, the rate of the movement is determined by the basic nature of the analyte. The nature of the analyte is going to decide its distribution between mobile phase and stationary phase. And the term that describes this rate is defined as KD, that is distribution coefficient, distribution between the stationary and mobile phase. In practice, the principle of distribution process may be used on that of adsorption, partition, chiral, ion exchange, or molecular exclusion chromatography. Analyte movement is going to cease only if the mobile phase reaches the end of the layer or capillary action flow stops or ceases or when the plate is removed from the mobile phase reservoir. So movement of the analyte is going to depend on its interaction with the stationary phase, how strongly it is going to get adsorbed onto the stationary phase. It will be retained more. If it is going to interact with the mobile phase more, it is going to run faster. So if we look into these terminologies, this is chromatography plate. 1 centimeter distance from the below is to be left before you mark the point where you are going to load your sample or spot the sample. So this orange color is the sample loaded. Now, once you put this loaded plate into the chromatography chamber, already mobile phase is in it. The mobile chamber is saturated with the uh, solvent vapors. The system is closed and the solvent is going to migrate upwards by capillary action. Once this run has completed from one end to the other end of the plate, you have to remove this plate and dry it. But very well you can see the spot where you had loaded your sample, that is the origin, the front, and the solvent front tin bin where the mobile phase has run. So you are going to have two marks, which are you are going to mark with the pencil or the marker on the back side of the paper. That is the origin point and the solvent front. And the separated spots, as you can see, pink here and orange here, are the two different analytes, and a third one is here, that is blue in color. So you are going to see these different sample analytes separated, and the distance traveled by each is to be measured. So distance traveled by the solvent front and the distance traveled by individual analyte is to be noted. The ratio of these two is going to give you retention factor or RF value, which is a ratio of distance traveled by analyte from origin upon distance traveled by solvent front from the origin. So, in terms of symbols, it is given as RF equal to DF upon DA. F is for solvent front and A stands for analyte, the distance migrated by the analyte. So, DF upon DA. Now, second factor that is to be talked about is K prime, which is given by the formula 1 minus RF upon RF. So, taking one example, numerical, we'll get to know about its importance. So, for example, in a chromatographic analysis of low molecular weight acids, say like butyric acid, that elutes with a retention time of 7.63 minutes. The column's void volume or time is 0.31 minute. Calculate the capacity factor for butyric acid. That is how it is talked about. Now, this is with reference to the column chromatography. But when it is with reference to the TLC plate, K prime is given by dF upon dA. What is dF upon dA? dF upon dA is the dF is the distance traveled by the solvent front and dA is the distance traveled by the analyte. So K prime is equal to 1 minus RF upon RF. Right? So the efficiency of thin layer plate can be calculated or observed or analyzed using these two factors. One is RF and another is K prime. Now, when it comes to chromatographic plate preparation, now as I said earlier, silica gel is commonly used. You have to prepare the slurry of silica gel, which is prepared either buffer in water or even in chloroform. The slurry is poured onto the plate and it is allowed to run down to the other end of the plate so that a uniform thin layer is prepared. However, preparation of slurry is a tricky technique and